Good afternoon, all of you. Today I am going to present a seminar on electron transfer theory. In this talk, I will start from microscopic complete description of the process. Then I will be discussing the theoretical methods involved in studying electron transfer. And then in the end, I will pose some open problems which will be further useful in studying them. So, charged complexes that are immersed in solution can exchange electron transfer, which are driven by the solvent fluctuations. There are two types of electron transfer. So, if the participating reactant complexes share a very small interaction in between them for a slight, a small electronic orbital overlap, then they can exchange electron transfer without affecting the inner sphere. For example, formation of ferricyanide from ferrocyanide in presence of small electronic overlap is an outer sphere electron transfer reaction. On the other hand, if the reacting complexes involve significant interaction, then one can see that there is a significant ionic movement uh, that happens during the electron transfer process. In this process, the movement of the substituting ligand actually mediates the electron transfer process. And as the inner sphere is affected during this reaction, this is called inner sphere electron transfer reactions. So in this talk, we will be primarily focusing on outer sphere electron transfer theory. This animation shows the dynamics of an electron transfer reaction. Here we can see that the donor molecules and the acceptor molecules comes towards each other uh, uh, until an approach distance. So once they reach an approach distance, they reorganize themselves inside the solvent in order to form what is called activated complex. So once the after the activated complex has been formed, the electron transfer happens. So after the electron transfer process, again these molecules reorganize themselves in the solvents. So it is important to note that uh, the molecules undergo solvent reorganization twice, once before the activated complex and once after the formation of activation complex. In order to completely describe the system, we start from the Hamiltonian description, accounting all energy contributions from the kinetic energy of electrons, kinetic energy of nucleus, uh, electron-electron interaction, electron-nucleus interaction, nucleus-nucleus interaction, the energy contributions from the solvent and the solvent species interaction. So we transform our reference frame from lab fixed origin to the center of mass of the molecule in order to resolve the dynamics into the physical motion of translational motion, translational coordinates, electronic motion, row vibrational motion, and into solvent motion. So the total Hamiltonian has been resolved by this transformation. So the aim is to study the eigenstate of the total Hamiltonian. Given the adiabaticity in the nuclear motion and in the solvent motion, one can express the total wave function as electronic states as a parametric function of nuclear and solvent coordinate. So this expansion leads to a set of Bonoeponomer energy surfaces. So here this expression gives the energy surface expression for the energy surface. This gives the equation of motion for the Lth electronic surface, equation of motion for the nucleus. As the chosen basis will not completely diagonalize the total Hamiltonian, this Lth electronic surface will be coupled to other electronic surfaces, which is given by whose strength is given by this non-adiabatic coupling operators. So if one does not choose the electronic state basis, then there would be a modification in the equation of motion with a modified non-adiabatic coupling operator. Based on these two different choices of basis expansion, uh, one can have two different representation of the same process. So if one chooses an electronic state basis, given the adiabaticity, it will completely it will almost diagonalize the uh, Hamiltonian. And given the adiabaticity, the motion of the nucleus actually will couple the initial and final state of the electron transfer process. Whereas in the diabatic representation, the owing to that the choice of the basis, there is no adiabaticity in the choice of basis. The nucleus will not couple the initial state to the final state. So one can also express the total wave function in as a linear combination of initial and final state of the electron transfer process. And in the diabatic representation, one can model the transition between initial and final state using this Hamiltonian description. So here one can see that the initial and final states are coupled through electronic Hamiltonian electronic coupling in the diabatic representation. In the adiabatic representation, uh, the motion of the nuclei couples the initial and final states of the molecule. So far we have seen that the electron transfer process is actually a curve crossing process. In earlier days, it was believed that this curve crossing was similar, analogous to that of the Frank Condon transition. So it was argued that the mass of the electron was very small in compared to that of the solvation atmosphere. So it was expected that the electron would immediately transit from initial state to final state without much change in the solvation atmosphere. So it was expected that as there would be not much change in the solvation coordinate, it was expected similar to that of Frank Condon excitation. So this posed a barrier to the problem. Uh, this energy barrier was calculated as the extra energy needed to add an electron to the acceptor molecule to that of its original potential electrostatic potential energy, and the solvation energy barrier was calculated. Here R would represent the radius of the ionic sphere, and epsilon R would represent the dielectric constant of the medium, dielectric constant of the medium. So the solvation energy barrier has been calculated as that of as given by this expression. Uh, and based on the molecular implications on this barrier expression, several predictions were made. So according to this expression, uh, a simple ions would would face a high barrier. So this explained that why simple ions take high time for electron transfer process, contrary to the expected. It also explained it also predicted that in presence of catalytic ligands, the solvation atmosphere of donor and acceptor molecules would be very much similar, so that the solvation energy barrier will be reduced. So he also explained the catalytic observation of the catalytic behavior of ligands. And he also predicted that for symmetric reactions, even small 0 point energy oscillations can overcome this Frank Condon barrier. He also quantitatively evidenced the electron transfer process using a simple uh, hydrogen-like model. And then Libby also argued that as this process is in the absence of any external perturbation, he argued that the introduction of the solvation barrier will be counterbalanced by the ionization energy reduction due to solvent. However, the conservation of energy for smaller time is violated, since this is a process in absence of external excitations. Initially, the energy conservation uh, is violated in this description. So this is the simplistic hydrogen-like model uh, using which Libby evidenced the electron exchange. So he evidenced the frequency of electron exchange quantitatively by using simple hydrogen-like 3D orbitals. However, Marcus immediately realized that the energy conservation was violated in the Frank-Condon explanation of electron transfer process. Marcus argued that reactants are characterized by some electronic wave function characteristic over electronic coordinates, and products are also characterized by their similar electronic wave functions. So the whole electron transfer process is moving from this electronic wave function characteristic to this electronic wave function characteristic. In order that, and it is also required that he also said that the activation complex will be a superposition of this reactant and products wave function. So in order that these reactants, products, and activated complex are all of eigenfunction of the same total Hamiltonian, uh, it must restrict that the energy of product state, reactant state, and activated state should be the same. So this argument let him explain the electron transfer process given by this schematic representation, which is different from that of the representation given by Libby. So during an electron transfer process, the reactant will be at minima initially, and and the product also will be at minima. And during the transit, the energy of reactant and product should match. So having discussed the scheme of the process, Marcus proceeds to calculate the reaction coordinate and free energy surfaces in order to quantitatively study the rate of the electron transfer process. So the reaction coordinate was taken to be so once the donor and acceptor molecules approach each other, the reaction coordinate they were fixed. The reaction coordinate was further proposed as the solvation energy difference between the initial and final state of the molecule. Here dp and dr would represent the electric displacement vectors due to reactant and product sphere respectively, and p of r will represent the polarization function. An alternative simple definition is the energy difference between the donor and acceptor molecules. So using these definitions of a reaction coordinate, uh, he proposes this reaction coordinate, and then uh, he proceeds to calculate the free energy surfaces of this process. So he assumed that the free energy surface of the process has a harmonic dependence over the reaction coordinate, and then he further calculates the free energy surface, uh, which constricts to the energy conservation requirement. So he optimized this Lagrangian functional, and he obtains the free energy surface for the reactants. So here we see that the reactants free energy surface is a harmonic surface, which contains spring constant as the solvent reorganization energy. So this solvent reorganization energy is described as a in terms of optical permittivity, static permittivity of the solvent atmosphere, and as the function of reactant complex and distance between the reactants. So similarly, he proceeds to calculate the free energy surface of the products as well, and then he obtains the free energy surface of the products. So this leads to the schematic description of an electron transfer process. Here one can find that the minima of the reactance is at zero. So as the solvent reorganization happens twice, once before the activation complex and once after the activated complex. We can see that the product's potential energy surface is placed at displaced twice from the solvent reorganization energy. And in order for the reactant to attain products energy difference, products configuration, the solvent reorganization energy should be applied on the reactant molecule. So in order to calculate the transition, uh, as said by the Marcus, the transition is localized to a single point. So in order to calculate the transition between the reactant and products. He proceeds to calculate the intersection between these free energy surfaces, and the intersection point was calculated to be delta G plus lambda x. Lambda x would represent the solvent reorganization energy, and minus delta G would represent the free energy released during the reaction. So based on the signs of this XC, one can characterize the process into inverted barrierless and normal process. So if the free energy released during the process exactly matches that of the solvent reorganization, the energy required for the process, then the process is a barrierless process, and then the reactants would spontaneously would start to form products. If the free energy released is less than that of the solvent less than that of the required energy, then there has to be some external energy in order to climb this barrier. Similarly for the inverted region. So for a symmetric outer sphere reaction, the delta G should is 0. So owing to high transfer at the crossing point, one can expect that in the barrierless process, the rate is very high, and in the normal region and inverted region, the rate is very low. So the rate was calculated using the Fermi's golden rule, and using the knowledge of free energy surface, the equilibrium distribution was calculated, and the rate was calculated as given by this expression. So so far we calculated in the diabatic limit. In the adiabatic limit, the rate of the electron transfer process is given by what is called Eyring's equation. So Marcus knew that the electron transfer process is driven by solvent fluctuations, which should be treated non in a non-equilibrium way. But he used equilibrium partition function calculation to derive the rate of the electron transfer. So, on the other hand, Sussman considered the non-equilibrium solvent fluctuations, and he used Smoluchowski equation to represent the non-equilibrium motion, and he calculated the rate of the electron transfer with a different pre-exponential factor. Uh, so both of these rate expressions consider only the single reactant state. However, Joshua Jortner considered both the states explicitly, and using a time dependent path integral formalism, he calculated an quite generic electron transfer rate. So from this expression, if the coupling between, if the non-adiabatic coupling is very large, it is in the adiabatic limit, then it goes to the Sussman's expression, and in presence of very less coupling, as that in the Marcus limit, so this expression goes to the Marcus expression. So this is a quite general expression which yields Sussman and Marcus expression as special limits. So as already discussed, uh, so if the so in the barrierless process, the rate should be very high, and in the normal process, rate should be low. But Marcus also predicted uh, less rate in the inverted region as well, due to which is arising due to the energy conservation requirement actually. So once the rate has been predicted, they were compared against the experimental data. So here we can see that in the barrierless region and in the normal region, the Marcus prediction is good enough to fit the experimental data. But in the inverted region, Marcus rate prediction undermines the rate actually. So this decrease in this anomalous result in the inverted region is due to the one-dimensional description combined with the energy conservation requirement. Uh, so in this experiment, actually, other degrees of freedom were not freezed. So the Marcus one-dimensional description is not valid enough, and then it was also observed that the rate constant had a high significant dependence on the uh, vibrational degrees of freedom. So in order to test the Marcus limit, the reactants were kept at fixed approach distance, and then now the one-dimensional Marcus prediction exactly matched that of the experimental result. So as in order to account the anomalous behavior of rate in the inverted region, so one has to also consider the dynamics along the vibrational coordinates as well, because the role of vibrational modes are also significant in the inverted region. So the two-dimensional potential energy surface is shown in this way, and the solvent relaxation is much faster in comparison with the vibrational relaxation. So this is the energy contour diagram of this problem. So reactants are characterized as one minima of the potential surface, and the products are characterized as minima of the other potential surface, and then they intersect. They can exchange population in this intersection region. So as because of the difference in the time scales in the solvent coordinate and the vibrational coordinate, one can consider a effective one-dimensional model, where one can calculate the steepest descent energy and then consider a one-dimensional problem with an intersection given by a parabola. So after reducing this two-dimensional model into an effective one-dimensional model, the dynamics of solvent coordinate is actually can be treated as a random process. So in order to treat the dynamics of dynamics of the solvent coordinates, the Langevin equation is used in presence of external and solvent forces. So if the solvent forces are assumed to have Gaussian white noise properties, then an equation of motion can be derived for the probability distribution over solvent coordinate over time. And then so using this formalism, one can calculate the flux probability of crossing the intersection region, which would eventually form the which reaches the activated complex, which would eventually is assumed to form the products. So using this formalism, one can calculate the rate, and this sinc function is actually chosen to be of the Eyring's form, where this intersection is calculated to be the parabola as shown in the previous slide. So far we considered that the electron transfer process happens from reactant to a single product state. So if there are multiple possible product states in presence of high frequency vibrational modes of the products, the problem is actually a multi-state problem, and the potential energy surface in presence of all solvent coordinate vibrational coordinate is given by a multidimensional harmonic oscillator, and subsequently all other excited states also can be given by similar formula. So in order to treat the problem completely realistically, one has to solve a multidimensional problem which accommodates dynamics from every possible degrees of freedoms. And then here we see that the population the population of the reactants and the population of the products are coupled. They exchange population, and the time evolution of this probability distribution is actually governed by diffusive Smolchowski operator, and this time dependence is introduced for the non-Markovianity. So solving such models can be very much useful in understanding can be used to understand several processes. For example, an electron transfer process is a basis for catabolism and anabolism in plants and animal cells, and then the electron transfer theory can be useful in understanding working of electrochemical cells. They are also useful in identifying reaction mechanisms in several organic reactions. So they can also be useful to study an interesting example of fluorescent blinking, where a molecule can go to its fluorescent state by accepting and by giving an electron, and it can lose an electron and go to a fluorescent quenched state. So this is called fluorescent blinking, and theory of electron transfer can actually useful in studying this process, and they can also be useful in understanding photo-induced electron transfer process, such models, the models which I showed earlier. So for references, this is the original paper of Rudolf Marcus, in which he initially treated the electron transfer process, and this is the original paper by Libby. And this book contains a very good review over the available methods in electron transfer. I would like to thank my supervisor, Dr. Anirudh Chakraborty, for introducing me into various fields, uh, so that I learned several interesting fields. And also would like to thank my juniors, Somesh Chamoli, Saurav Kumar, for their valuable teachings and discussions in chemistry. And I would like to thank you for the time spent. Thanks.